Lactose is a disaccharide sugar that is converted by E. coli into galactose and glucose, which are used by the cell as sources of energy. To make use of lactose, the bacterium must first transport the sugar into the cell and then split it into galactose and glucose. 3 genes are involved in the utilization of lactose. These are called LAC-Z, LAC-Y, and LAC-A. LAC-Y codes for the enzyme that transports lactose into the cell. This enzyme is called the lactose permease. Once inside the cell, the lactose is split by the combined action of beta-galactosidase and beta-galactoside transacetylase, coded for by the LAC-Z and LAC-A genes. These 3 genes form an operon. They're placed next to one another in the E. coli chromosome, with a single promoter just before LAC-Z and a single terminator signal after LAC-A. This means that the 3 genes are transcribed into a single mRNA. When there is no lactose present, E. coli does not need to make the 3 lactose utilization enzymes, and the operon is not transcribed into mRNA. It is not transcribed because a regulatory protein, called the lactose repressor, is bound to the DNA close to the promoter. The site at which the repressor is bound is called the operator. When the repressor is bound to the operator, the RNA polymerase cannot get at the promoter. This prevents transcription of the lactose operon and ensures that the enzymes for lactose utilization are not made when they are not needed. Now we will see what happens when E. coli finds a supply of lactose. Although the operon is switched off, the cells always contain 1 or 2 copies of the lactose utilization enzymes. These are enough to transport 1 or 2 lactose molecules into the cell and split these into glucose and galactose. A byproduct of this reaction is the compound called allolactose, which is a disaccharide with a similar structure to lactose. Allolactose binds to the lactose repressor. This changes the shape of the repressor protein, and subsequently it is no longer able to attach to the DNA. Once it detaches from the operator site, RNA polymerase can bind to the promoter and transcribe the lactose operon. The bacterium can now take up the rest of the lactose molecules and convert all of them into galactose and glucose. Gradually, the lactose is used up. As the lactose is used up, the amount of allolactose byproduct gradually declines because the bacterium also converts the allolactose into glucose and galactose. Eventually, there is no allolactose left. In the absence of allolactose, the lactose repressor is able to bind to the operator once again, blocking the RNA polymerase. The lactose operon is now switched off until the bacterium finds another supply of lactose.